computer. Bingo. Okay. All right. Well, welcome everybody, and welcome Simon to this little AGWB Club Night special virtual edition in the good company of Froth Technologies. Simon, thanks so much for coming along. Looking forward to hearing from you as this it was a couple of years ago we met with you before, and it was a great little talk about your good work at Froth. That was back in the lockdown days. Since then, recently, you've released your homebrew pouches and offerings, and it would be great to hear about those. And also, a couple of weeks ago, we did some tastings of 3 of the beers that use the Wilding yeast. So again, that would be good to find out more about the background to that and discuss a bit of that. So awesome. Over to you.
Over to me. Yeah, well, welcome everyone. Thanks for inviting me in back again for another chat. Yeah, it was lockdown when we were last had our one of these, wasn't it? Yeah, cool. So I think from our emails, Clive, we were thinking that we could talk a bit about the Wilding yeast and also the homebrew yeast pouches which we've developed. What should we start with?
Up to you, my friend. Wherever you like.
Alrighty. Well, given that your lips are still wet with the Wilding tasting, I'll just go kick off with that. Now, yeah, maybe I can just give you a little bit of you guys an overview of sort of how we approach that, because it has been a multi-year project for us pretty much since we started. We've had that dream of like, yeah, cool, these domesticated yeasts are great, but can we find something wild and from New Zealand out there? And yeah, do shout out if you have any questions along the way. Just put that on mute. Yeah, cool. So when in the early days of Froth Technologies starting out, we had this dream to sort of find some wild yeast that would bring a bit of flavor as a medium for storytelling and the grain to glass story of beer in New Zealand. No doubt some of you folks have tried some interesting wild fermented beers out there, and you can, I suppose, wine has taken a lot of the glory in terms of the term terroir and what like a bioregional signature can bring to a food or a beverage. And so actually microbes play a big part in terms of the terroir of wine as well. So yeah, our dream was to get out there and see if we could find our own wild yeast. We decided we wanted to find Saccharomyces cerevisiae, so ale beer yeast. There's 1,500 known species of yeast out there, so all from the get-go, we're looking for just one. And then, of course, not all of them are going to be great fermenters or, you know, great performers in the brewery. And so with that in mind, you really want to start out with a lot of samples and a lot of isolates, because a lot of what you find isn't going to be any good. So what that looked like for us was we wanted to get away from the cities, away from breweries and people and cities and get into the bush and try and find some unique microclimates and native plants and see if we could find some yeast living on those. So early on, we had a chat with Callaghan Innovation about this. Myself and my co-founder, Ryan, we're both very much from a brewing background. So we've kind of, just like yourselves, home brewers that started to muck around with commercial brewing a bit and actually put our heads together where I was doing some contract brewing under the label Kakariki Beer Company. Ryan was actually brewing my recipes at a brewery previously known as Eagle in Christchurch. He sort of shut himself down. And yeah, around that time, we'd thought about actually starting a craft brewery together, as you do. Probably thought that at some point. But our market research said, let's start a yeast company instead. Like there's a bit of a gap there in terms of sourcing local ingredients in New Zealand. So yeah, our background is beer, home brewing, but when it comes to some of the more nitty gritty research and development, the deeper science, a lot of our day-to-day is self-taught. So we went and saw the guys at Callaghan Innovation, and together we kind of developed this kind of yeast bioprospecting bootcamp process that took a lot of their scientific knowledge and our kind of goals, dreams, needs for what we wanted and needed a yeast to do, and developed this program together. And from there, we needed to collect a bunch of wild samples. So we went to different parts of the country. We went up to Northland, deep into some of the forests there. We actually went to, I don't know if you guys have heard of Fire Dragon chilies. They grow super hot peppers up the far north and make super hot sauces out of them. So we wanted to be, quite mindful about what land we were setting foot on. We did look at stepping onto dockland, but that was a whole kettle of fish and like actually thousands of dollars of paperwork so we sort of stopped that very early on and thought, who are our mates in different parts of the country who just happen to have a backyard that's like full of bush. Clint at Fire Dragon Chilies was like, yeah, bro, come go up the back bush and you can poke around in there. So yeah, that was our first stop. We also went to Hawke's Bay and sort of snooped around some of the orchards. We also went to the west coast of the South Island and also the Rangitikei district, sort of central North Island. And yeah, we were basically just looking to gather a bunch of aseptic samples, berries, bark, soil, and also some ear traps. So basically like a mini cool ship, just basically a jar of boiled wort left out overnight with a mesh on top to sort of keep the bugs and bits out of there. And then the next day you go fetch it and it starts fermenting with a mixed culture. And yeah, and so those samples, we took 45 individual samples total, and they went to Callaghan as a first step, and they sort of enriched them into a mixed culture, so like a real zoo of bacteria and yeast at that stage. They were then able to isolate out individual yeasts and sort of start selecting for brewer's yeast at that early stage, using antibiotics and stuff to inhibit bacterial growth. And yeah, it just started to build up this library of yeast isolates that are most likely brewing yeast, yet to be confirmed. And so that became a library of 400 individual isolates from 45 samples.
Just to interrupt you. So how do they know it's brewer's yeast? Because it looks like brewer's yeast, right? You're looking at it through a microscope and you think, so you're trying to work out the particular cells that kind of look the part. Is that
Yeah, good question. At that stage, there's a few things they're doing. So yeah, some of the selective processes are like antibiotics to go, okay, cool, that's not bacteria, it's yeast, and then plating them out onto petri dishes, agar plates with certain selective inhibitors on there that for example, this plate should, like wild yeast shouldn't grow, but Saccharomyces cerevisiae should. And so it's still not like bulletproof, like it's still not PCR confirmed, but you're just, doing rough kind of weaning out the wild, or, the non-sac strains, and honing in on, by that stage, it's like, these are probably brewing yeast isolates. But yes, you're also right. They also would do that morphology assessment of like, okay, it certainly looks like brewer's yeast on the agar plate as well. That sort of round, ovoid, white, shiny, compared to like fluffy or bumpy or other ways that other yeasts can look.
Sounds like quite an expensive process. So did you actually manage to get any sort of grants or anything to cover this with kind of an innovation or anything? Or
yeah, it is sort of frustratingly expensive, and I suppose if we were to do it again, it would be cheaper, because we sort of would fast track it in how much we've learned through developing this process. So yes, we did get a project grant which sort of covered half the costs through each stage, but yeah, it was still about like 60 grand all up from start to finish to build this library. And I suppose wilding is the first one to be commercialized out of that library. There's still more there sleeping in the cryobank that we sort of wanted to give wilding a good go, and when maybe some of the interest dies down, we could pull out, start to look for the next wilding, essentially. Yeah. And so from that stage, like I say, we've got 400 isolates. We're still trying to whittle them down and work out which ones are useful in a brewing setting. And so they used some clever sort of fast throughput boot camp analysis there. We decided that we wanted to see which of these strains could ferment maltose. And also we wanted them to have a tolerance for different strengths of alcohol and also pH. So Callahan had sort of said, if you start with like basically dosing the fermentation with a certain ABV, and then the yeast keeps growing, you can sort of safely assume that yeast is comfortable producing. We did 5% ABV and 7% ABV as our two strengths, and then a pH drop. And then the wort was just sort of like a lab version of just like a malt-based starter, essentially. And so they have some really nice tech there where you've got a whole bunch of little wells that'll have just like 2 mils in each, and each one gets dosed with inoculated with a different yeast strain. And then it goes into a machine that can kind of fire a beam of light through the wells and measure cell density before and during and after, and sort of 6-hour increments. So basically measures if the yeast has grown in population, so it's happy and healthy. And so through that we're able to get a metric of like which of the strains didn't mind the higher alcohol, was okay with the pH drop, was okay with maltose fermentation, but we still don't know. We still don't have any idea if they smell or taste any good, or if ultimately if they're Saccharomyces cerevisiae. And so these stages are taking sort of months and months as well. We're sort of probably into year 2 by now. It took us a good probably 6 months just in visiting those different wild parts of the country. Callahan needing time to build out the library, one sample at a time. But we sort of whittled that 400 down to about 93 promising performers, which still we're not getting any readout on like attenuation yet. We're just showing growth happened. So the but at least we've whittled out 300 that were just didn't do much at all. And so sort of from that 93, some further analysis took place. There was some more plating where we were trying to there were some different approaches for trying to work out if we had brewer's yeast yet or not. But we sort of wound up at a stop-go point where the next stage was looking just very expensive, where they were sort of like, oh, we will just PCR the whole library. So we'd be, paying a lot for.
Tell me what PCR is. Sorry, Simon. PCR.
Oh yeah, it's just a technology that you've probably come across it with COVID and that, but it's
right. Yeah, it's that. Right.
But yeah, it's just a way to sort of sequence the genome and to be able to see for sure what this species is based on its DNA, essentially.
Yeah.
It's just an expensive process. So at that point, we actually decided, well, hey, we've learned enough about these strains. Let's do some in-house testing, and if we find something that we're interested in and that's a keeper, let's send that for PCR testing, and then find out what that is for sure before we start offering it to breweries and that. And so we were sort of running through batches now at the sort of 1 litre fermentation, benchtop fermentation trials in our laboratory. And in one of those first batches, I mean, to be honest, we're seeing a lot that there's interesting characteristics in terms of sensory. Interesting esters and floral characters and phenolics, but sort of attenuation typically conks out around like 2 or 3%. But we did have one of the early ones stand out in the bunch, and sort of cross-checked our database back to where that one had come from. And that was a sample from the Rangitikei district, an air trap. So again, that boiled wort in a jar with a mesh over it, a mini coolship. And that one had actually also gone from that starter and had done a full homebrew batch of beer. And so actually the sample that went to Callaghan was like a finished bottle of, like a swing top bottle from the cellar. So, I mean, I guess you could say that's one of our predictions in terms of helping to find quality yeast, is like, rather than going straight from like berry or bark sample to wort and just trying to isolate everything that's in there, do a brew first, and that's going to kind of whittle out the microbes that aren't up to the task, and the strong ones that survive through the complete fermentation, the hops, the alcohol, and settle into your bottle. The ones that are alive at that point are the ones that you actually want. And so that's what happened with the yeast that we wound up releasing as Wilding. And by the time we'd sent that away for testing, confirmed it's Saccharomyces cerevisiae with the diastatic gene, which is that unique trait of all wild sac yeast, that it can excrete an enzyme that can turn some of your lesser fermentable sugars, maltodextrose and things like that, snip them up into simple sugars and ferment your beer extra dry. That drying of the fermentation just tends to happen sort of, we see it as like primary fermentation in week 1, and then week 2 you'll get those last, like it'll go from, let's say 1008 down to 1002. That's typically where Wilding's finishing.
That explains what we saw when some of the beers we brewed. Sean did one in particular, which I don't know if he's going to unmute his mic, but Did you ended up at 999, didn't you, Sean?
Unmuting now, Reese.
Yeah.
Yeah. And there you go. I'll turn on video, Simon, so you can.
And it certainly it can do more than 7%.
Yeah, so a commercial brewer took a sample of my finished brew and took it to his alcohol tester, and it was 8.5%.
Wow. Yeah. Yeah, I think in-house testing, we got up to 11% comfortably. And we do have one distillery in Whanganui. They're a very small one called Good Bones. They actually sort of substitute about a third of their grist with old bread from the bakery and birthday cakes and baked goods. So they use less grain. They've used wilding to make rum, and I'm not sure what strength they were going up to, but yeah, it's amazing. How's the mouthfeel on that beer? Because I mean, on paper, it's exceptionally dry.
Yeah, it certainly doesn't taste like it's 8.5%, but once you finish the glass, you know it's 8.5%.
Yeah, and still got some of that mouthfeel from the Gossamer production. Yeah, cool. Yeah, so we knew we had something interesting because we're sort of now sniffing and tasting the early samples of the yeast that became known as wilding, and to the horror of Callaghan innovation, they're like, no, you can't just sip samples of brews that you've inoculated from the wild. I'm like, have you been to the wild workshop? Like, the whole place goes like that. It's like, well, no, we're pretty comfortable if the pH is below 4.5 and the alcohol's over 5. Like, pretty happy tasting that.
Well, it can only make you stronger, right?
I think so. Yeah, just thinking about ancestors. But yeah, we sort of ran it through some more like profiling, I suppose. We did different fermentation temperatures, different pitch rates, and what else? Yeah, just to see what it does. Because this is sure, it's got character that maybe would call farmhousey or Belgian, but this is a strain that's just been happily evolving in this valley in New Zealand for who knows how long. So we didn't want to put any boundaries around it. We just wanted to see what it could do. And yeah, quite kind of interesting that it didn't necessarily like thrive or do anything more necessarily beneficial or at the higher temperatures. Like some of those Belgian strains, the more you wind it up and just let it free climb, the more it can get quite rustic and expressive. But yeah, we didn't sort of find that so much. We just sort of found like some lighter alcohols and sort of nail varnishy things starting to happen, which our sort of hypothesis there was, or I suppose summary was, well, New Zealand's a very tepid environment. So it's not like it would typically be seen like 27, 29 degrees out there in Hunterville. It would be, evolving in colder temperatures. So it was happier there. And then, some of the brewers at Emerson's had sort of sparked some interest around this strain along the way. We sort of bump into them at a beer festival and talk to different brewers. They've got some really, really quite skilled technical brewers there that have come out of the Otago Brewing School. So yeah, quite nerdy brewers as well. And yeah, they just had to convince Lion to be okay with them bringing a diastatic strain into the production brewery. But so they actually had to, they did a secret trial brew in their pilot brewery, which is actually 1,200 litre batches. I know it doesn't sound like a pilot brewery, but their main brewery is 5,000 litre brew house. And yeah, they did this sort of brew that you could only get at their tap room. It was called Frothy. And I did have a few brewers who were just like other commercial brewers stopping through Dunedin and say, hey, what's this brew? It says frothy. It's awesome. It's like this farmhousey thing with this kind of lager-like finish. So that was actually the first brew, but Emerson's was just trying to give it a go before they went and scaled it up. And from there, they did. I don't think they changed the recipe at all. And
so what can the diastatic do in a brewery then?
Yes. So the main fear for a brewery is that brewery tanks are cleaned very well. They have, I don't know if you've seen the spray balls that exist inside them, basically a sprinkler inside a tank that blasts different detergents and sanitizers, runs down the walls of the tank and dumps out the bottom. So in theory, it's like hitting every surface, but sometimes there can be, just things that can be missed, little pockets or little shadows that the sprayer doesn't quite hit. Not normally a huge problem, but with a diastatic yeast being introduced into a brewery, it only takes just like a handful of cells in the wrong place, like a clean beer, to outcompete that clean beer, or even in finished beer, even though the clean yeast has done the job, the beer's finished, it's at its final gravity, no more fermentation's happening, it's ready to go into cans. If that got contaminated with even a tiny bit of diastatic yeast, that yeast can excrete an enzyme and turn those unfermentable sugars into fermentable sugars and cause extra fermentation to happen, which we welcome if we're using the yeast intentionally. But if that happens in a brewery tank, one day you've got a nice clean pale ale, and then you come back and you're like, what the heck? It's now at 1002 and it's kind of funky. I can't sell this as my core range anymore. Or if you miss that and it's contaminated going into cans, that re-fermentation can happen at the supermarket. And that actually did happen in the United States, where I can't actually remember the brewery, but they had cans exploding up and down the country, being stored at ambient, which were now, popping from that re-fermentation. So, it does make some breweries a little bit nervous, but it's totally fine. Like the Wild Workshop Garage Project makes both clean and very, very funky wild beers in the same tanks. You just got to be good at it. It's totally possible if you're paying attention to your hygiene. But Emerson's actually wound up sterile filtering that first wilding brew in order to sort of get the sign off from line, but basically before allowing that liquid to go into their canning line. And so a sterile filter is basically just like has a fine enough filtration that every single yeast cell is fermented out. So the final beer is completely free of yeast.
Is that why their beer was particularly clean tasting?
Yep, that would certainly be part of the reason. They also, I think they're like keg beer poured with a lot more of a robust head, while their canned version had a more dissipating head. They said that was sort of frustratingly from passing well carbonated beer through that mesh. It's sort of just knocking out some of the CO2 along the way. But for sure, if you filter something down to that level, it's gonna take, particularly like some of the hot particles or polyphenols, that super clean. But we're glad to get them over the line and to have the first commercial beer brewed with that wilding yeast. And they put together a really cool label for the can as well that I think told that story well. But has anyone else done a homebrew with that yeast?
It was just me and you, wasn't it, Reese?
Yeah, well, you gave me your slurry.
Yeah.
Uh-huh.
Hey, remember how mine was like my brew was like a New England IPA wort, right, with oats and wheat and stuff like that. How it looked quite like it. Well, I've just poured the last of that mini keg that I brought along to last club night. I don't know whether you can see it, but look how clear it is now.
Yeah,
It's
Interesting.
almost looking like the Emerson's, eh?
yeah.
Was that with the wilding yeast?
Yeah.
Interesting. Yeah. So you were saying it was looking quite hazy.
Yeah, yeah, because
Yeah.
it's a New England IPA wort that I
Uh-huh.
did, so it has oats and wheat
Nice.
in there.
That's cool. Yeah, it's certainly slow to flocculate, and any yeast that's slow to flocculate is quite frustrating when you're trying to produce it in a yeast lab. You know, we need it to populate and then drop so we can harvest it and package it. So yeah, we actually have Wilding has its own dedicated tank at our facility purely for the same reason of like if Wilding wound up emanating some, jugs of vape that have gone out to some breweries, it could have the same accidental funky hazies everywhere, which would not be, we just can't have that happening. So we actually bought a one of those 100-liter tanks. I think, does the brew shop have them? They basically just look like two kegs welded together in a tall tower. One of those keg menter, on wheels. You're like, hey, but that's also allowed us. We've just done a batch of French Saison, custom propagated for some breweries where, we've had them in our cryo bank, but we've never propagated them up for. We just didn't want to put diastatic strains into our main tanks. So it's cool to have a funk tank now. And we just grew some of that up for Kitadu, and also some went to Australia, which is cool. Looking forward to doing some.
An export license for that, or an import license, Aussie style?
Beer yeast is a approved microbe, so it's on the sort of safe list along with, bread yeast, beer yeast. They're like, oh, we're not worried about that. You just have to very clearly state that on our paperwork.
But how are you going to stop the Aussies claiming it as their own?
Yeah, exactly. Yeah. Well, they were very quick once we started sort of telling this story over here. Some of the brewers over there looking at our social media and going, great, love it, Wilding, like envious. You Kiwis have got your own wild yeast to play with. That's cool.
Yeah.
Do you want to come over here and help us? Like, yeah, we have one brewery. All expenses paid. We'll get you over and you'll find us a yeast. And it doesn't. It's not like we can just knock that together in a weekend. Like, we're 3 years deep and 60 grand of, project funding here.
Yeah.
I hope you got deep pockets, but no, they did want to get their hands on their own ones. But
hey Simon, is that the issue with the diastatic strains? Is that mitigated through pasteurization, or is that like just some breweries that pasteurize their beers, would that mitigated, or is it worse than that?
No, pasteurization would do it. There's just not too many breweries that have pasteurizers. But yes, no, that would be a way to ensure you didn't have any can or bottle bombs.
So I guess Emerson's don't pasteurize their beers then?
No, they don't pasteurize. They centrifuge. If you're familiar with that, it's sort of that spins out almost every particle in a continuous kind of whirlpooling filtration pathway. But not everything. Like almost all of the yeast will be removed, which is always an example I use when sometimes you have a brewer who's like, oh, my hazy's dropping bright. What's up with the yeast? Like as if the yeast has sort of settled out of their beer and now it's see-through. It's like, well, actually, that haze should be coming from your proteins from your malt and your hot polyphenols permanently binding together. Emerson's brews all their hazies with our vape strain, and they'll centrifuge them so all the yeast is gone. So you should be able to like filter a beer or crash cool it, condition it, let all the yeast drop out, and it should still be hazy. Like you shouldn't be relying on yeast particles to do that. But Emerson's did have to bring in a sterile filtration for that. It just depends on I don't know how much risk or how much throughput or how much shelf life they're looking for. Like pasteurization and filtration does add shelf life, which can be really helpful for export. I know Castle's was sterile filtering their beers. And like McCashin's does have a tunnel pasteurizer along with B Studio. Yeah, but they're not super typical. Mostly just rough and ready. Yeah. Any more questions about wilding?
Like, have you measured the sort of like glycerol levels? We sort of found in beers we tried the glycerol was still leaving like in the sort of 5% beers. This was the typical mouthfeel you might get out of like a typical saison yeast. We sort of found that Sean's definitely a better mouthfeel at 8.5. We sort of felt like that sort of 7% is where 6.5 to 7% is where that maybe that mouthfeel is going to come through with that wilding yeast.
Oh, cool. No, we haven't actually. That's not something we've looked for. We sort of, I suppose by the stage, by the point of having something that was remotely ready for commercialization, we were just itching to get it out there. And it's just super interesting to hear folks like yourselves trying it out and doing on the ground research, more or less. Like I think that Emerson's wilding, what was that, 6.2% or something? So still not quite at that 7 mark, but that's really interesting. You're saying the 7% starts to really show off some interesting mouthfeel.
Yeah,
Well, we sort of made that a
Sean's was-
guess because it was sort of like a 6.2. Like typically with those sort of wild yeasts that go really, really low, they produce enough glycerol that sort of adds that mouthfeel back in, even that it's really, really dry. We sort of found it with the Sabbath warding is really, really nice. And it was an older can, so I think it was from the initial brew. Now you're talking about months and months later, so it's like it's had an evolution over time, but it didn't have the typical glycerol that you typically get out of like a saison yeast. So sort of potentially a higher alcohol beer would sort of add that sort of mouthfeel back in, even if it's going super dry.
Yes. Yep. Yeah, no, that's a fair assumption. We also sort of find it overall, it's not the most like rustic of characters as well. Like it's sort of quite which that actually sort of mostly that excited us a lot from the get-go, because we knew that a wild yeast, they can be all kinds of quite sort of funky and all over the show, but we didn't want to necessarily find something that was so like rough or strange
and approachable, sort of
approachable. And so we've had all these ideas about trying to tell the story of like all New Zealand ingredients, New Zealand wild yeast, but if it was just super strange, and I've actually just been blown away by how approachable that beer has been. Like I actually got a 50-litre keg of it for my brother's wedding, and I was serving it myself, just pouring glasses at the bar, and aunties and grandmas and granddads and some people that normally wouldn't touch craft beer at all were like coming back for seconds and going, that's a really lovely brew. They don't know necessarily what they love about it because they're, not at the nerdy level. Yeah, but they're like, if it was too hoppy or too bitter or too funky or weird or something, they you'd see unfinished glasses around it, but they were just like, and so I just thought that was really cool. I was like, yeah, you're right. It's not the like most rustic, robust sort of farmhouse strain out there, but I reckon it's a great first one to have out there because of, I'd like to think of it as a transitional, like bring people into the funk a bit more gently and go, because it's also not sour. Some people are very like sour phobic, like can't do them. But yeah, cool. What other wilding brews did you?
The beer Baroness one, the table beer.
Oh, that was what, Three Sisters.
Three Sisters, yes. 3.8%, I think it was.
Nice.
Yeah.
Yeah. That was a cool one actually. Joe had been dreaming up kind of a New Zealand saison with some herb additions. I think he was thinking like manuka and horopito and some New Zealand forage botanicals. I think he maybe that one didn't go ahead. He was trying to think of.
No, we did try the one with horopito because we noted that the horopito was kind of on the label, but it didn't have that pepperiness that we expected.
Oh, was that the
Renaissance?
That was
Fortune Favors Bushwalk.
Yes.
Yep. Yeah, so they kind of went with that. Yeah, it sort of had that Captain Cooker Manuka thing going on, but I suppose the fermentation profiles, well, it's not as showcased with those other additives, but sort of nicely propped it up, I thought.
It was good beer.
Yeah. But yeah, that Three Sisters one was an interesting take. It sort of suggested that to Joe as we'd sort of seen some table beers out there emerging, and I'd seen some examples more in the US of folks sort of dry hopping them with some new age hops. And he was like, that sounds good. We'll do it with Citra. And that beer's done quite well, actually. I think also in the Australian Beer Awards and also New World Top 30, which we thought was super cool because, like I say, going back to wilding, approachable, not too weird. This is still a wild yeast in the New World Top 30. Like, I think that's pretty cool.
Yeah.
Yeah. Any more questions?
Was it all the like the different strains that you've kind of got to the point where you've got a few of them that are possibly candidates? Was there certain I guess common environmental factors at play? Like were they taken from similar sorts of environments, or are they just all over the place?
They're actually all over the place, eh? Yeah. I mean, we would, I suppose, like ascribing our human bias to it, we'd find like harakeke, flaxbush, all the tui are just like going berserk around it. They're also like dripping with nectar, and you're like, oh, this feels like a yeasty environment, but and then you get the results back because some of our samples had zero growth whatsoever, and then others would have like 15 isolates from one,
So was it all the same time of year?
Say again?
Was it all the same time of year?
No. So yeah, we do have everything logged, like down to the day, the time, the weather, the temperature, the GPS. It's all there. So I guess once
go back if you wanted to.
Yeah, I guess we're still looking for some more success stories so we can look back upon what worked. But yeah, the strongest.
Could you find a straight Saccharomyces amongst these that doesn't have the diastatic side of things, do you think? Or is it unlikely?
As far as I know, all wild yeast has that diastatic gene, and it's been bred out through domestication. You can also, I mean, you could, you could breed it out in a lab. You could genetically modify it if you weren't in New Zealand.
Or you could feed it enough white bread from your other mate, and that will get rid of it.
Yeah. But yeah, I mean, because like Lallemand yeast, for example, has released they've got their farmhouse strain, which is a non-diastatic
the Philly sour.
farmhouse. No, I think it's called farmhouse is the name. It's a hybrid yeast. And so they have put two different parent strains to sort of forced mating in stressful conditions where they're forced to sporulate and make hybrid yeast, or many hybrid yeasts, and they sort of look through the bunch and find the ones that still holds the traits that they're looking for. So it'll be like, cool, it's got the saison characteristics, but it's lacking the diastatic gene from this parent. So that's a way to kind of to breed in specific traits without GMO, because at the moment, they're still quite strict here in New Zealand. It's definitely something we've looked at. In Canada, Escarpment Labs has done some really great strain development there, where they'll genetically modify two yeast parents, get them to breed, and then sort of turn that off again in the offspring. And so in Canada, and I think pretty soon Australia, they consider the kind of next generation, even though they had GMO parents, they're considering the offspring to not count as GMO, as part of like relaxing the laws around it in certain areas, like brewing and breed yeast seems to be one of the early stepping stones of, like, we're not talking about mammals and stuff, we're just talking about a single celled thing and making it like not diastatic, like it's very simple function. But no, we're not quite there yet.
Do you need to go there if you've got this point of difference and it's New Zealand and it's from the wild? Do you actually need to go that GMO route?
Honestly, it's just a case of we've had a fair few brewers asking for it. So it's this whole like thiolized yeast trend. Where these certain yeasts can convert thiols into other compounds, and basically can sort of get more profile, different profile out of your hops by interacting with the yeast, right? It's just sort of naturally it's very low detection for humans. So this is about just like dialing right up. Like One Drop in Australia has done some, that brewery has done some that I tried I think at Biavana, that were just like a whole nother level of wacky, crazy hoppiness. But it's still hard for them to get it. We have had some New Zealand commercial brewers ask for it. Like, can you, I don't know, I guess they're just chasing the latest trend and trying to make the next dank beer in those cases. And so I mean, we've looked into it in the sense of like, what would it take for us to do that? But yeah, no, you're right, we are pretty happy with our New Zealand wild yeast that we've managed to find as a needle in the haystack. And yeah, hopefully we can find some more in there as well. They are just, they're certainly hard to come by. But I do think that once they've been through a brew, the odds are a lot more in your favor. We actually did a little project with Garage Project where we went to the Botanic Gardens here in Wellington, and with the beekeeper, and they opened up their beehives, and we were sort of swabbing all different parts of the hive, taking all these samples of like tickling bees and poking into a honey cell. And
I thought honey had that sort of bacteria growth inhibiting kind of stuff in and around it. That
Yeah, it's mostly just like the density of the sugar and how they the bees are able to keep the hive so dry. You know,
Right.
it's like that osmotic, yeah, just being like, yeah, when sugar is that dense, it's actually just like and inhibits bacterial growth because it's too dense. Like if you get
Right.
some water into your jar of honey, or your moisture, it'll start to ferment on the top, because now it's been diluted. But yeah, no, we didn't find we couldn't find anything useful and out of all those hive samples. So yeah, it just goes to show, just like no matter how fun the project is or how cool the story is, because Garage Project was like, yeah, we're gonna make what we could make some kind of beer for bees week,
Ragged, is it?
something like that, and use some honey from those hives, and do something with some wild yeast at the wild workshop. But yeah, we couldn't find anything. Well, I mean, yeah, we'd get a little bit of fermentation, but nothing much useful for a proper brew. But hey, there's a lot more like these low and non-alcohol beers out there which actually call for a yeast that doesn't do much fermentation. It sort of can't simulate all those sugars. So yeah, maybe we could revisit the library and with a different lens, and yeah, be looking for something like that. Yeah.
So have you been looking at the lows and zero alcohols as a?
It's just sort of stayed in the back of our mind. Like we've sort of tried to stay focused on our goal, even as along the way it was like, well, these are non-sec isolates, and we're like, oh, well, what are they? It's like, well, we can find out for you, but we have to carve off another section of the project and get more funding. And it's like, oh, okay, no, let's stay focused. We're trying to find brewers yeast that can ferment beer to a reasonable strength in a brewery. Okay, let's just keep going along that goal. And so
Sounds like you need to find a PhD student.
right, you get a bit lost along the way.
Yeah. Someone who's prepared to drink their own water. Yeah.
Yeah.
Oh, good on you, Simon. Yeah, no, it's an amazing story. It is an amazing story. Very cool. Well, yeah, very cool.
That must be super challenging. Sorry, that must be super challenging knowing that, most of the yeast, if you do find a yeast that is going to survive those conditions, it's probably going to be diastatic. And knowing that you're trying to sell this to commercial breweries, knowing that they're going to be really skeptical about a diastatic yeast, it doesn't matter how good it is. Like, how does that play with the mind when you're like literally trying to find a yeast that's probably have that strain?
Yeah, no, that's totally a thing. And I think, I don't know, so much has changed since we were starting out on this journey. I mean, yeah, when we first approached Callaghan, we were like, yeah, cool. We're going to find New Zealand yeast and New Zealand breweries will make beer with it. Simple as that. But we've just, yeah, over time and throughout this project, we've come to learn and realize that actually, yeah, well, for one, not everyone is, in terms of the beer drinkers out there, going to be keen on a saison, even if it's a kind of cleaner leaning one. Not every brewer is going to be keen to introduce it to their brew house, and even if they do, they're not going to be like, cool, that's part of my core range now. We have sold pitchers of yeast, of wilding to a number of breweries now, and it's been really cool to see what they dream up with it. But they're not super likely to crop yeast from it and do another brew. Three Sisters is probably an exception because they placed in the New World Top 30 and they had to make a bunch more. So I don't know, maybe it's, hey, maybe the times are changing a little bit. Maybe if more people did want to drink saisons and farmhouse beers, then brewers would certainly, the brewers do want to make more of them. It's just what the punters want to drink. And so, no, yeah, you're right. It is a little bit frustrating sort of by we're getting to the later stages of the project going, I hope somebody actually wants to buy some of this at some point, because there's no way we're not likely to recover all our costs, but damn, it's a cool story.
Hopefully coming into summer, you might start to see a bit more of those lighter,
Yeah,
more interesting beers.
totally.
Do you think like the more scientific approach that you took through Callaghan Innovation,
Mm-hmm.
do you think like moving forward you'd continue to apply that much rigor to it, or do you think you'd loosen the reins a bit? And
I think we'd probably start by going just back to our existing bank, that cryobank of library of strains that we've got, and go straight back to our like in-house benchtop 1 litre fermentations with sensory in the office. But yeah, if we were to go back to the drawing board, I think it's certainly helpful to have that scientific approach or scientific detail at a later stage. Very much like just brewing a beer or a starter from start to finish, inoculating it with a good amount of wild microbes. So, not just like a single berry, but like a branch or, really just like whack it. And so it's like really strongly inoculated with a population, and it's just the survival of the fittest. Let it complete a proper fermentation and then isolate out of that. I just think it puts you about 5 steps ahead of, and eliminates those sort of first 5 steps that we had to sort of wade through initially. So yeah, I'd bypass that next time. Yeah.
If you want to put something in some, send up some samples, we'll propagate it and report back if you like. We'll spread it amongst the masses and see what happens.
Nice.
Bust the cool ship out.
Yeah. Well, given that Craftwork have a cool ship that they push outside in their alleyway that little blue penguins walk past, and that's how they make some of their beer. So, I think it depends how big a balls you got, really.
Totally. And I mean, yeah, that was another thing we considered along the way was, maybe we should have I think we were just hooked on the idea of seeing what's out there in the bush, But yes, it would have been, maybe we would have made a bit more progress early on if we'd say got a bottle of something from the Wild Workshop, a bottle of something from Craftwork, and just let's see what's in the bottom of these bottles, because these have obviously proven their track record. They've done the dash.
Yeah, but not as cool, mate. Not as cool.
Not as cool. Oh, we could just do this, the spontaneous isolate series. But I think the Wild Workshop they were meant to be firing up their cool ship today, but there was a shortage of raw wheat. So it's been pushed slightly. We don't get any nights here that are cold enough, because their cool ship's 5,000 liters. It's shallow. It looks like a kiddie pool, but it needs to get I think below like 5 degrees overnight for just that volume of work to cool down to a low enough temperature for the wild microbes of Taranaki Street to blow in there.
So this is still your day job then, is it?
Yep. Myself and Ryan are in there and at our yeast lab full time. Yeah.
Did you find that the homebrew pouches sort of helped with that at all? Because obviously, any business when you first get it going, it's always a bit tough. You got to pay the bills. Is a yeast patch has actually taken off enough to help with those brews?
Yeah, for sure. It's I mean, there's been some more like ups and downs for the craft brewery industry in New Zealand lately. Or some of our customers have gone out of business, or some of them are struggling. So it's but yeah, the homebrew stuff has certainly sort of filled some of the gaps and seen a bit more consistency month on month. Homebrewers, as you know, are quite social consumers. Here we are chatting about it online. And so naturally there's a good amount of word of mouth, and we've just love hearing about some of the brews that have been made, some of the feedback, some of the experiences, and then brewers telling other brewers, hey, I've given this a go and here's my results. So I mean, we've wanted to supply to homebrewers since the get-go. It's just been sort of head down and trying to produce yeast well at all, and consistently, and supplying New Zealand breweries to pay the bills. But it has been more like technical and difficult in terms of taking a living yeast culture that we want it to ferment, we want it to produce CO2, but not when it's in a pouch. Like pause that for a second, go to the homebrew shop. Okay, now you can, now that you're out of the pouch, you can go back to fermenting. It's like, so we've talked with some other yeast labs overseas about that, and picked up some sort of trade secrets and stuff. But
Have you talked to White Labs at all?
Still a work in progress. Not White Labs lately. Escarpment Labs we stay in touch with. But there's been, I mean, so much of our business in our yeast lab in Lower Hutt has been trial and error. Like I say, we came from a brewing background, but a lot of just like homebrew thinking has been applied into, here's what we need to do. How are we going to do it? And sometimes it's solved with homebrew equipment as well. Like our yeast packaging tank is one of those SS Brew Tech bright tanks. I know there's sort of the uni tanks are more conical, uni tanks are more common, but we picked up one of those dish bottom bright tanks and fitted a stirring arm on it and some legs and wheels, and we collect our concentrated yeast slurry into that, and it's stirred as 100 liters of creamy slurry, and from there that's what we were doing earlier this morning. We're packaging off a big batch of Buddy and
So you're starting with DME, or are you starting with wort?
We start with liquid malt extract from
the old
from
school
down
Maltexo.
down to Dunedin, and we actually have our own sort of special brew done there, because we sort of get 8 drums at a time, 200 litre, 320 kilo drums. So
is that out of the Stoke plant, is it?
The what, sorry?
Where's the who makes the Black Rock cans?
Yes, it's that. It's I think it's in the Spates,
the original
warehouse, and within that is like Maltexo / Black Rock spray, concentrated malt extract production. And so we get a hopped malt extract, highly fermented.
You're using hopped for your propagation.
Yep. Yeah, we had been dosing that ourselves. It's hop extract. We had been dosing that ourselves in-house, but I've just found it easier to have it arrive already at the specific IBUs we need.
But that's to inhibit other things, is it? That's the idea of having it
bacterial growth. Also just sort of because our finished product as well winds up as well, it's yeast slurry and some spent wort. We discard most of the, probably 90% of the spent wort, but naturally even though that yeast slurry is at about 2 billion cells per mil concentration when we package it, naturally it still, flocculates and separates out in the chiller. And so it's nice to have that, blank sort of protective blanket of liquid over our slurry. I think it's about 5% alcohol, and so having 10 IBUs in there helps, prevent spoilage as well. Even though we are actually sterilizing our wort in house, we boil it at 121 degrees.
It's under vacuum then, under pressure, is it?
Under pressure, yeah. So it all gets loaded in, and it's got a steering arm in there, and then the hatch gets closed, and it comes up to 100. And then as the pressure starts to climb to 15 psi, the temperature climbs up to 120.
Sounds like you need a turbo wastegate or something on it.
Yeah, it is. Well, yeah, as soon as you sort of, it's quite a spooky kettle. Like it's got a glass window in the hatch with thick glass, so you can look inside. But because it's not exposed to atmospheric pressure, it's actually still in there, so it doesn't rumble around like your kettle does. But as soon as we're done sort of cooking it, it's only a 10-15 minute at 121 degrees. As soon as you start venting that pressure, it automatically erupts into a boil all of a sudden. But and yeah, makes a whole lot of steam. But yeah, we'll sort of do a big batch of that in a week and just leave the hatch closed, and from that we actually do it high gravity at 100, and we will dose in a certain amount of that into a tank and then follow it through with sterile water. We've got a sterile water system, so it just means we can sort of flush the pipes and the hoses and dilute our high gravity wort into a tank. It also means we don't need as big a kettle,
so you can just draw off as you need, sort of thing.
Yeah, we'll be able to feed like a 100-litre tank and a 1,000-litre tank off of the one little 500-litre kettle.
Oh, so you build up into the 100 and then into 1,000.
Yeah, they sort of have 24 hours at each stage. So prior to that, it's in a little 15-litre propagator, and then 100 and then 1,000, 24 hours apart. So yeah, we just feed them straight out of that sterile tank. Yeah.
5% 10 IBU, you could sell that on Courtney Place on Saturday night to a few punters.
We have distilled it.
International lager.
That's a good one to watch, actually. I don't know if you've ever caught Jesse from Chase the Craft. He does a home distilling YouTube channel. It's pretty cool. He came and visited us and took away 20 litres each of the spent wort beer, 5% 10 IBUs from Rude Boy and Buddy and Vape, and he distilled them separately to make whiskey, and then sort of did some rapid aging on some oak, and then tasted them side by side. And I've tasted them, and quite interesting how different the different yeasts are into the final whiskey.
Oh, really? You can taste it through?
Yeah. Because we're doing quite like rough and ready brews. Like we're aerating the brew the whole time through those 24 hours, so it's constantly getting rumbled with sterile air. So it makes, for a pretty oxidized brew, but it also it's quite estery, and yeah, it's not pleasant to drink on its own. But I think distilling fermentations, the they like it rough. They like sort of fermenting hot on the grain. Like the more like rough it is, the more character you get in the final distillate. Like they'll distill on the yeast slurry and all this whack stuff that we normally wouldn't do. Yeah,
Well,
they sort of abuse
Was
it.
he the one that did the distilled the fermented Mountain Dew?
Yes, that guy. Big beard,
Yeah,
Palmerston North. Used to work
did
at
all
a
that
dog
stuff with
food
Clawhammer.
factory. Yes, correct. And now he's a full-time YouTuber and he does pretty well from it. So, he just makes great videos. Even if you're not a distiller, it's just quite interesting to.
I'll have to find some links and add them, Clive.
He's done some with beer as well, like distilling IPAs, or he distilled Corona and then put it back into Corona to make like a fortified 12%
Imperial Corona.
Imperial Corona, yeah.
You'd have
Stuff like
to call
that.
it COVID, wouldn't you?
Yeah. But yeah, he knows his stuff.
In terms of the yeast strains you got at the moment, is there some that are like, obviously Wilding, we're talking about that now, but is there any other yeast strains that sort of taken off? You're talking about Rude Boy and Vape and Crispy and stuff like that. Is there any that have been more popular than others?
They sort of seem to go through different like waves of popularity, we did sort of, one thing we found out when we started our business was we're like, cool, we need this range of yeast that brewers use. We need an American ale strain, something sort of more traditionally English, hazy strain, definitely like a lager or two. But we sort of found out pretty quick that it's I don't know why the lack of access to yeast strains in the past, but a lot of commercial brewers have not been using lager yeast for their lagers. In some cases, you'll have a can that says lager right on the front, but it's brewed with USO5, or a lot of New Zealand pilsners brewed with USO5. So it's kind of like, maybe it just a nod to that style where maybe a New Zealand pilsner is kind of an XPA with a kind of pilsner grain bill. Like, who says it isn't? So we found lager to be a bit of a slow start, but over time we're seeing that's picked up more and more. And
so with your pitch rates, the lager would show it more than anything, wouldn't it?
Yeah, you're right. And that has been another thing we've helped brewers with, is just sort of, there's certainly been just a case of I put 2 blocks in. I haven't really thought much about what that pitch rate is, or I haven't adjusted for. Yeah, so we've sort of helped polish up some really good pitch rates, and it works. Like, the likes of some of these breweries that have won trophies and gold medals, little small breweries in different parts of the country, a lot of people haven't heard of. They're just nailing their fermentation. Like, sure, there's recipe and there's process and stuff, but I mean, if you ask the brewers, they're like, we get, like, Three Sisters will tell you it's because we get fresh yeast and we outsource all our yeast management to Froth, so we don't have to worry about that part. We just focus on making good beers. But we got another lager yeast strain, Vagabond, by request of Garage Project. That's sort
Can
of
you say how you source your new strains?
Yeah, we just initially get in a pure culture, usually from a yeast lab or a bank or wherever we need to get it from in a pure form. It could come as a slant, or maybe frozen culture, or
so you pay for that sort of thing as a commercial sort of thing, or is it just because you just buy a sample and then you scale it up?
I'm sure it's something that anyone could get their hands on. We then sort of inoculate it into our own, lab propagation, and then so grow up a healthy culture in-house, and then get it ready to be stored and frozen in our cryobank at -80. So that's just a case of sort of like, loading it up with the right nutrition and getting it into a kind of antifreeze type refrigerant so that it can go into -80 and just go dormant and not just break open the cell walls
when you rehydrate it.
Yeah, pretty much. And so all our stuff that's in the cryobank, we almost never touch it. Like, it's very much the mother stock. It's like it's got its own backup battery. It's where everything sort of starts from. But typically, like, our day-to-day yeast production, like from that cryobank sample, it's going on to an agar slant, and that'll kind of be in our lab fridge for I don't know, I'm not actually sure how long, but it's probably like several months at a time, 6 months, longer. And so new batches are started from just a few loopfuls off of that slant. And so only once that slant is spent, like several slants will be made at the same time. So it's probably only like once or twice a year that cryo freezer gets opened to replenish our working stock on agar. Yeah, it's kind of like the backup of the backup of the backup.
Mm.
Yeah.
We're going to rely on you for that when the what the hell they call it when the world ends.
Yeah. Yeah. The time
Armageddon.
capsule.
Armageddon, yeah.
Armageddon. Yeah.
When we want to start all growing our own grain and hops, and people go, "Fuck, where are we going to get the yeast from?"
Yeah. Or repopulate Earth, and it will just start with yeast cultures.
Yeah.
Yeah, I've had a good go with the vape. That's worked well. Yeah.
Okay.
It does finally clear eventually, weeks later it'll come clear.
Okay. You're finding that you're getting it to drop bright.
Yeah, well, this was a vape. That was vape. Yeah.
Okay.
Not quite bright, not bright like Sean's, but yeah, brighter than it was.
Mm-hmm.
Yeah. I mean,
Is that after packaging, or is it still on the yeast at that point?
after packaging. Yeah. I mean, it's out of the keg.
Mm-hmm. Yeah, we can see that that's shown up for commercial breweries as well, which is super frustrating for the whole team. The sales peeps are like, our hazy's see-through. Do something, brewer. And he's like, what do I do?
Stop making hazies. That's what you do.
Yeah. But one of the main ones has been nutrition around that. So when I say nutrition, I mean mostly like free amino nitrogen, but also oxygen. And so like these yeasts have the ability to excrete an enzyme called protease, which is like, if the yeast is sort of malnourished, it can release that enzyme, and it's able to metabolize your protein haze as food. So the yeast kind of like out of desperation gobbles up your haze. So we actually import and stock a yeast nutrient called yeast mode. We get it in because we need it for our yeast propagations, but we've since started offering it to breweries, and they love it. And in some cases we've had breweries with clear hazies fix that problem just by switching to that nutrient. So trying to think what's
is that in the powder form?
It is, yeah. I think there's a similar one called Cervomyces that you can get at a homebrew scale. It comes in capsules.
Yeah, that's the White Labs one.
It's the White Labs. That's an active one, as in it's actually like living yeast cells. So you chuck it in your kettle to to bust them open. But our one is a yeast extract, so it's like the insides of yeast cells, not the cell walls. They've been step one for making Marmite. Yeah,
Right.
yeast extract. So it's high in that free amino nitrogen fan content, which is especially good for because hazies, you're taking out barley, which already has a natural fan content, and replacing it with oats. And then maybe winding up the gravity. And you're using a yeast strain which is English, top fermenting, loves oxygen. So we've even seen breweries like they'll have their row of tanks right in the brewery, and then there's the brew house and the heat exchanger where they boil the wort, cool it down, send it to a tank. If the tank is closer, they'll use a short hose to connect it. If the tank's further away, they pull out the long hose and hook it up. And they've found in some cases you'll have the close tank is dropping bright, the long tank not, because through the heat exchanger they're oxygenating their wort ready to pitch the yeast. But the one that's traveling longer down the hose, it's all bumping around and mixing in there and getting better dissolved oxygen, and therefore better hazy fermentation. So they've solved the haze retention just by using a long hose, even if it's the short tank, if you get what I mean. Just little like low-hanging fruit like that, or you try one or try the other. And I mean, nutrients are a real easy one. You just and not all nutrients the same. So I do mean that kind of like organic nitrogen based ones can really be super helpful for haze retention. Aside from, I mean, we do actually have a one-pager document on tips for brewing hazies, but not everyone's gonna need to check that out. But honestly, it's funny. You'll have maybe a year or two ago, it was like, how do I make a hazy? Was what the brewers were saying. Or how do I make permanent haze? And since then, we've seen some commercial brewers like sort of borrowing techniques from brewing hazies, and I mean like mid-ferment dry hopping, like double dry hopping, maybe putting some higher protein stuff in their grist, but maybe there's some oats, maybe there's some wheat, and then winding up with a beer that's meant to be bright, like an IPA, going hazy, even if they've used, like an American ale strain, and they're wondering why. Like, we're not talking like a real milky, cloudy hazy, but a definite, permanent haze that they cannot filter out, cannot fine out, and they're now ringing me up going, how do I get rid of my haze? I'm like, I thought you wanted haze. So, all sorts of things. Well, basically, it boiled down to like, you look at the recipe and like, it looks like you're trying to brew a hazy. Like, you've got wheat, you've got oats, you've done mid-ferment dry hopping. It's like, yeah, but I used American ale yeast. It's like, so that's, that strain's got some haze positive components to it. You sort of borrowed some of those juicy techniques and you got juicy. Don't know what to tell you. Or the other one is contact time. Three Sisters told me about that. They got a new hop cannon where they sort of, load it into this vessel and circulate sort of dry hop separately. But what were they finding? That they used to have no trouble at all with haze stability. But since using the hop cannon, they're able to sort of like, I suppose, dry hop the beer so rapidly and get all of those, all of the hoppiness extracted into the brew, shut it off. But they were finding it wasn't actually enough contact time to really bake in that permanent haze. So they're like, it needs to actually rest on those dry hops long enough to kind of, for that haze to stick around. And vice versa with their clear beers, just like leaving that, getting carried away with the mid-ferment dry hops. And it's funny. Chasing our tails sometimes.
Yeah, funny you say that. Actually, because I've had hazies that have stayed hazy, like milky hazy for that 6 months, and I don't put nutrient with most of my yeasts, but there's a couple of yeasts that I do, and those are the ones that stay hazy for a very, very long time.
Yeah.
And I tried to a West Coast with one of these strains, and it just I had to give it like 3 rounds of clarification to try to clear out. Like it really just didn't want to drop. So I'm just wondering if that nutrient or something in there.
Yeah. Yeah. Sounds like it, eh? What nutrient would you normally use?
I don't know the name of it handy, but yeah, I just thought it was super interesting. But I'm going to actually look
Yeah.
out after you mention that. I'm going to look
Uh,
out for the ones and see if it is high in that free amino nitrogen and stuff like that,
yeah.
because
Yeah.
that completely makes sense.
Yeah. That's a funny one, eh? Yeah, the yeast can gobble up that haze that you want to stick around, or yeah, it can unintentionally show up based on your recipe. So yeah, I've learned a lot about it over time, and it's just kind of funny sometimes answering these phone calls for sometimes brewers you've looked up to, a lot over the years, and they're picking up the phone, calling for some brew doctor help. It's like, okay, give me the vitals. All right. Because sometimes it's like I've got 10,000 liters of it and I don't want it to go down the drain. It's like, oh no, we can save it. We can save it. Here's how we'll fix it. Or,
Do you think it's
yeah.
also the fact that that they feel like if they speak to you, the secret's safe? It's just kind of like
Yeah.
you're not going to go tell every man and his dog. Whereas if they went to ask another brewery, it'd be like, oh, this brewery asked me this. It's like, why is this brewery asking me this? Right? Could be a
Yeah.
little bit of that.
For sure. For sure. And I do find myself in a position of, I suppose just sort of being a middleman where I'm not, usually I'm not naming breweries to other breweries. I'll just be like, along the way, I've learned this, or I know that another brewery tried this and it worked well. You could try that. And yeah, usually if something's like a real trade secret, a brewery's pretty forthcoming about that. They'll be like, hey, by the way, this is, and that's not honestly not that often, because our breweries here are pretty relaxed and collaborative. Yeah, I had 3 sacks of oat hulls dropped off to me yesterday at the East Lab from Kedadu Brewery because Garage Project needed to borrow some for that spontaneous brew. It's just like super, turbid, thick mash. So
Kiruru seem to be doing quite well recently, I reckon. Beers recently seem to be quite a notch above what they used to be, I think.
Yeah. I sort of found them to be a little bit restrained in the past, like held back on sort of ingredients. Some of their offerings just hadn't quite nailed the punch that some of their competitors had. But yeah, no, there's been some exceptional stuff. I mean, their barrel program is pretty top notch, but yeah, also top shelf. What did you try recently?
Oh, I can't recall actually, to be fair. At the beer spot with 40 taps, you kind of
Get lost in them.
then you got to remember. So.
Yeah.
So the newest one we've got here, which I believe using your products, St. Leonard's.
St. Leonard's. Yeah. You guys in there. They did a fresh hop with wilding. I haven't actually tried that. I try to get my hands on all of them, but sometimes it's like if they're not doing rigger fills, or you have to go try it in person. But Panhead also did one out of their little pilot brewery, which they were doing riggers of online. I think it's batch 283. It's like a Belgian pale ale with wilding. I thought that one was exceptional. Like they sort of didn't hold back on the bitterness and the kind of caramel malt.
Yeah, well, you'd almost expect a clash with bitterness, and that doesn't seem to happen with
Yeah.
Kind of.
Yeah, it's just like real like funky sort of like marmalade-y. And yeah, I found myself sipping it and going, oh, I just love the kind of the yeast character with that caramel sweetness. But then I'm like, wait, this is like 1002. There's no sweetness here. So
We
perceived
found
sweetness.
with some of the older home brews that we've tasted, we've kept a bit, that Wilding over time sort of developed sort of a honey note.
Oh, okay.
And what was it that Kelly was picking up? He kind of got a, wasn't a minty thing, it was something like that.
Yeah, no, it was getting mint. Yeah, mint was like common right throughout.
But that was palate,
honey,
though, right?
was it peach? Was it peach that was coming through on a regular?
Huh. Cool.
There was definitely mint.
This is like after it was like 4 months old or something like that. 4 or 5 months.
Cool.
Hmm.
Nice. Oh, that's awesome. Yeah, there was a brewery in Queenstown did a brew as well, called Farmhand Canyon Brewery, Queenstown. And they use sort of like malt from their farm, honey from their hives, and foraged elderflower and dog rose, which I think is kind of like a wild rose, climbing rose that has little sort of hibiscus type buds before they open, which I think that's what I was tasting. It was kind of like it's almost sort of like smacking sort of astringent, like slightly hibiscusy type.
But was that still a hop beer?
Say again?
Was that still a hopped beer?
Yep. Yeah, it was. But yeah, quite cool, that sort of expression of the local area, that terroir factor, and then with the wild strain on top. I thought it was done quite well.
Yeah, when I do a rebrew of what I did for that end of year thing, I'll send you a bottle this year. So
oh, great.
It was an interpretation of a Norwegian farmhouse ale, but using totally Aotearoa ingredients. So I swapped juniper for manuka.
Yeah.
Used home smoked for Hutakawa, and no hops. So that was, and Pilsner malt, but with the Hutakawa smoke and the Wilding yeast. So I was very happy this year because I got to use a New Zealand yeast with it.
Awesome.
And that was around the 6.5%, and which I think is a sweet spot. So
nice one.
I'll send you a bottle this year.
Great. Yeah, it's just so cool to see what folks come up with, because there's no sort of like Wilding is this style strain. It's like, that's why I've been sort of nudging brewers to try out different stuff as well. Like, okay, with the Belgian Pale Ale, it's like, okay, who wants to do something with some real bitterness now? Who wants to do something more session strength, quaffable spirits? Like, what's next? My dad actually, my dad's a home brewer in Christchurch, and so he's sort of, well, I don't know, maybe he always outsources his recipe writing to me because maybe it's just easy. But I think I just grabbed him like the Gladfield XPA recipe and then just fudged it around. Usually he's like, I've got these hops still in the fridge from the last brew. So I'm like, cool. Switch them around. Boom. There's your New Zealand XPA, but done with Wilding. I think
did it go dry though? Did it get down to 2003, 2002, or was it
wouldn't. I don't think it would have gone that far. It also had like a little bit of I guess it must have been a yeast haze to it. He had it on keg, but just like almost like a soft kind of powdery, like Hogarden type haze,
Okay.
I've been trying to convince a brewer to do a wit with it, because I have found like some of the homebrew forums online, brewers forums, some folks saying, oh, saison yeast is my preferred yeast strain for doing wits. Like, yeast doesn't know or care what it's meant to be brewing. It's just got characteristics and history. So yeah, I don't know.
Coriander and orange peel sort of things.
Yeah, or substitute some. Sometimes brewers will use other, like Hallertau usually does on with tangerine with our unicorn yeast. Garage Project that has done a grapefruit and rhubarb wit the last couple of years, and they're about to do, what is it? Some like spiced, like heavily spiced wit, like I think like Indian spice, so like garam masala wit. But I was nudging them like, do it with wilding, but they were a bit chicken about bringing the diastaticus in. So but yeah, I'd love to see that done. I reckon it'll be a winner. Like, just a bit drier. Again, it's it's not the most like rustic of farmhouse strains, so I reckon it would suit a wit real well.
And sorry, what's the unicorn?
Oh, that's that's the Hogarden strain, Belgian wit. We do it seasonally. It's a shocker to make it. Like, we normally chuck it on, ferment it, like propagate it in our big tank, and then leave it cold, crashing over the Christmas New Year period, because it just, if we did it during our normal production, normally it's like, it sort of has a week in the big tank and it's all done and dusted, fermented, chilled over the weekend, and by the Wednesday it's all settled out and we're packaging it, and the next lot's going in the same day for the next batch. So as soon as you're waiting 3-4 weeks for one yeast strain, you got to pause everything else. So it just doesn't work.
Is it slow and steady, or what's?
No, it's just such a slow flocculator. Like, yeah, it'll be like 2 weeks deep. We'll take a sample and it's just still just sitting in there. It's just suspension. Like, maybe the top has gone bright, but it's just so slow to flocculate, and we need it to, otherwise we can't crop it out and send it away.
Would you be better off? Would you be better off just to have less? Would you be better off just to have like less yeast and just cut after a week, and just hope that you got enough flock, like you got some flock in a week? Or
yeah, we can do that. It's just about like a small, the next tank down is like a tenth of the volume, and it's also like, we will struggle to sell the whole batch. We'll do it at springtime. We were like, right, wit season, who's making wits? Like, Craftwork has done one, Hallertau, a few others, but yeah, we definitely don't sell it all out, which is a shame. Wits just aren't,
popular as they should be.
Not enough of them, for sure. And so we do still offer it throughout the year. We do like custom propagations. So the brewery will more like get a small starter pitch of a couple of liters, and they'll have to grow it up one stage in their brewery. But yeah, that allows us to, every now and then grow up a strain that's not part of our core range. If there's a particular brewery that's like, I actually want this. So Castles and McLeod's, they both have their own sort of secret strains that we grow for them, both lager and ale. But then stuff like French Saison, which we've just done, or we've just done a custom propagation of unicorn. I think it's actually still brewing away for Garage Project. We've grown that up in a smaller tank just for them, but still it'll be like, it's still going to take a couple of weeks in that small tank. It does drop out quicker because it's got less distance to go than a tall tank, tall tank's like 2.5 meters tall, but the wee keg is just like 0.5 meter.
With that, I was sort of thinking of those horizontal lager tanks, right?
Yeah.
Like,
Yeah,
I'm sort of
yeah.
getting those vibes. It's like, hey, why don't you just go horizontal lager tanks?
Yeah, or I was thinking we'll do a 100-liter tank and you can just take the bottom half of that tank in a keg. Like, do you mind, if it's got some of our spent wort in it? But yeah, just pitch the whole lot in. Don't wait for it to settle. Just chuck it all in.
I'm sure Twofold with their new horizontal tank will probably take it off your hands.
Right.
So they just cut up
Twofold.
and Parnell, Bernard from Excel.
Yes, he has been in touch actually. He's a true lager brewer. He won a trophy with the Vagabond yeast. So yeah, looking forward to getting him some. Also heard from old Mike Nielsen, who started Panhead. He's just bought Boneface Brewery. So that was cool. Feels good when, reputable brewers open a new brewery and they're like, first things first, let's talk to the yeast lab. So, yeah, thanks guys.
So how do you, because you can't copyright an organism and that sort of thing, so what's to stop someone taking a sample of the boiling yeast and then selling it overseas and calling it a different name and that sort of thing? Is there any way of protecting anything like that?
Yeah, you can patent a microorganism and you prove its novelty just by sequencing the DNA and checking it against a global database and going, this is new.
So this is a recent thing, or has it always been like this?
It's always been like this in New Zealand. In the States, you can't. But it's definitely kind of in the online, I suppose, like yeasty, milk the funk, virtual space. It seems to be generally frowned upon. And I don't know, our thinking around it is, our terms like froth and vape and buddy are all trademarked just as words in the yeast category. Mostly to I suppose just prevent some of the bigger breweries mucking around with our IP without permission. But what's to prevent it? Nothing much really. And I mean, even if we suspected some brewery was like storing, using, propagating, or sharing, or selling, that strain, it would just be a lot of resource to even prove it. Like we're sort of investigating brews on shelves and even just proving that like this brew was fermented with this strain, which is protected. You're just having this big legal battle that's no good for anyone when it's like, it's not like we're keeping that strain exclusive from anyone. Like it's so much easier for them to just come buy some.
I was thinking more rather than the breweries, more along the lines of, say, the White Labs or the the other kind of big lab banks, big yeast banks,
Yeah. I suppose we've just sort of taken shelter from the smallness and remoteness of New Zealand so far. But yeah, it's
is it something you can actually license back to them and get that kind of thing into the States where it's not protected by doing a licensing thing?
Yep. And that's something we've talked about, just like licensing or just like strain swapping. Hey, you've got some cool strains you've developed. We've got one strain that we're willing to share through bioprospecting. You open to strain swapping, and so we've definitely had those conversations. So I suppose just like the craft beer industry, you'd expect it to be a bit more like they just pick up the phone and be like, hey, wilding sounds cool. do you want to? Can we get a vial? How to, just be transparent about it rather than sneaky. So
Yeah.
Yeah, that's sort of what we've experienced so far. But haven't had a phone call from Chris White yet.
I've got his number.
It's on the yeast pack. 0800 Pac-Man.
No, I've got his cell number.
Nice. Yeah. Nice one. Well, it's been
All right.
awesome to hear about all your brews and experiences and
Yeah.
feedback in general. And
Hey, I said an hour and we've been 2, even though we started
Oosh.
a little late. So
nice.
I'll wrap up the recording aspect at least. So yeah, much appreciated. Thank you, Simon, for sharing
Thank you.
all of your insights. It is a fascinating story, and it is fantastic that we as home brewers get to brew with the yeast that you grow here in little old Aotearoa, New Zealand. So it is a very cool story, and good on you for pushing on.
Thank you.
Talk about that dream. Yeah, definitely feel as though you've had that dream and followed it and living it and breathing it. So yeah, it's great. Really
Thank
good.
you.
Much appreciated.